So my name is Catalina Cabrera. I am going to present today emphasis in sheep reproduction, but I'm going to cover a couple of other aspects of my research interest with all of you. So I joined the University of Nicosia a little bit less than a year ago, and I am very pleased to introduce some of the work that I have done in the past. So this is a brief outline of the things that I would like to cover during the seminar. I would like to first do a brief introduction about myself, and then when I start talking about some of the research aspects, I would like to cover some clinical cases, interesting clinical cases that I have seen and published. And then I'm going to go in a little bit more of emphasis in the area of sheep reproduction. After that, I would like to basically let you know a little bit more about how I can see that some of this knowledge can be used for future research. And then I will briefly go over some conclusions. So as a general introduction and information about my background, I grew up in Colombia, South America. I had the opportunity to work or be raised close to animals, and that most likely was the reason why I went into veterinary medicine. So I studied at the University Nacional of Colombia in Bogota with an interest in production animals. And while I was doing the degree, I started having some interest in the area of reproduction. So after graduation, I worked for a couple of years between Colombia and Uruguay. And at that point, I was working mostly with cattle in commercial facilities that were mostly using high genetic, high merit animals as donors for the collection of embryos. These embryos we will either freeze for exportation or transfer fresh, just with the idea to improve different herds. So after a couple of years doing this, I moved to California. I went to the University of Davis, California, and I did a master's in preventive veterinary medicine and a residency in production animal hair health and reproduction. So during this period of time, I did a research project was based on the area of embryo transfer. So it was factors that affected the production of embryos in cattle. And then after I completed the residency and the master's, I stayed in the veterinary hospital at the University of California, basically working in the hospital, visiting different farms, and mostly teaching fourth-year veterinary students. After that, I went to the University of Florida, where I continued to work with basically commercial operations, both in farm and at the hospital, and teaching veterinary students. So during this period of time between the two universities, I had the opportunity to see some interesting clinical cases, mostly in one of the areas of interest for me, that is congenital diseases or congenital malformations, mutations. And so I want to present to you some of these clinical cases that we were able to publish. So the first case that I would like to cover is this case of this miniature Highlander cow. This cow came to the hospital in the University of Davis because she was having an abnormal pregnancy. It was determined at this point that she was carrying an abnormal fetus. So we induced parturition, and eventually the animal went to C-section. And what we found in the C-section was this malformed fetus. This is called a bulldog calf because of the similarity with the bulldog dog breed. As you can see, there are several abnormalities in this fetus, including the cleft palate. I don't know if you can see my arrow here. So cleft palate, a shortener of the nostril, short legs. It actually had a lot of skeletal abnormalities. It had a spina bifida and absence of back legs, very short front legs. It has evisceration, and abdominal hernia. And then out of interest, I took the head of this calf, and did a CT in comparison with a normal one-day-old calf, just to basically evidence a little bit more of the abnormalities of this fetus. So this is just a brief comparison of the abnormalities. So a similar abnormality has been described in other breeds. We have not seen it in this breed, but there is a genetic test for it. So we decided to test both the cow and the bull. And what we found is that both of them, the male and the female, were carriers of this mutation. So this is an autosomal recessive gene. And basically the combination when these two carriers will transmit the autosomal recessive gene to the offspring is going to produce this bulldog calf that is basically a lethal gene. So basically the conclusion in this case was these two animals should not be bred again, since they are both carrier and we have a 25% chances of having this mutation. But also it was important to discover that this mutation is present in this breed. So this was reported for the first time. Here we have two cases of interest as well. This is a 1-year-old bull. This bull came to do a breeding soundness evaluation, and at the moment that we were evaluating the reproductive tract, we were able to notice that there was a abnormal development and basically an abnormal closure of the urethra at the tip of the penis. So in other words, the opening of the urethra should be here at the tip, but we can see that it starts a little bit earlier. So in this particular case, this bull had been used for production and it was being used for breeding cows. And because of the concerns of the possible inheritance of this malformation, it was recommended that this bull was not used any longer. So we were able to obtain tissue from it and actually confirm at this point in the histopathology that indeed these animals had a failure in the closure of the urethra. So this abnormality is called hypospadias. And in this particular case in the bull is, let's say, mild because it's only at the tip of the penis. But later on, we had another case that was a lamb, and you can, let me try to orient you in this picture. This is like the abdomen of this lamb, and what you can see is in this area where the rectum is located and the tip of the penis should be, there is a complete opening of the urethra. So all the external urethra of this lamb was exposed, had not closed during the development of the urinary tract. This animal actually, when we received it, was already post-mortem. It also had a lot of skeletal abnormalities. We did some CTs and some X-rays just to be able to evidence some of these abnormalities. We tested for a couple of non-genetic diseases, but they were all negative. So in this particular case, it was unknown the origin of these abnormalities. This animal was born to a twin, and the twin was normal. So that kind of rules out the possibility that it was exposure during pregnancy. So it could be still something genetic associated with this lamb. The recommendation for the producer was to keep an eye and follow any descendants of both the male and the female in case this is something that will repeat again in the future. So these two cases of hypospadias are relevant also in humans because it's one of the most common abnormalities found in humans, and there is still not a clear understanding of what are the factors that are associated with the abnormality. The last thing that I would like to show you is this case of this set of twins. These are two Brahman males that were born in Florida. And very soon during the first and second month of age, they started developing an increase in the lymph nodes. So at that point, we started investigating a little bit further, did some blood work and were able to determine that they both had leukosis. We were testing for bovine leukemia virus. They were both negatives, so they were their mother and sire. So we were able to rule this as the cause. Nevertheless, they were declining in health very rapidly and they were euthanized. And then in the following exams in histopathology, we were able to confirm that these both animals had leukosis that was later diagnosed as type A. So this lymphoma, basically in the twins, left the door open to try to understand if there was one of the twins that had the lymphoma and it was transmitted in utero to the other twin. None of these things could be confirmed, but it also opens the door to things that we need to understand better about the transmissions of lymphoma in utero, also because it's relevant for humans and pediatrics. Okay, so now I'm going to dive a little bit more into the area of sheep reproduction. I want to cover a little bit an overview of some of the aspects that might be relevant to go in more detail into the research that I have been done in these animals. So I will shortly share 2 research projects that were related with this topic, and then some of the findings and how this can be applied. So as a brief overview, and for those of you that are not familiar, small ruminants and other species have developed or have evolved into having a breeding season. So these species will not cycle year-round, they will just cycle for a period of time every year. And the interesting part is that regardless of how long the pregnancy of this species is, at the end of the story, when you look at the period of time where they give birth, this always coincides with the end of the winter and the spring. So in other words, these animals have evolved to make sure that they give birth in the spring when they are going to have more availability of food. So going into the detail of the specific species that we are talking, so it's more ruminants and more detailed sheep animals, we are going to see that, again, they usually start lambing in the late winter, spring, early summer, which means that their breeding season is basically in autumn or fall. So the way that these animals identify that it's time to get, to work and get pregnant is through their sensitivity to the hours of light and the presence of the stimulus of this light. So in other words, what happens is that these animals are going to be susceptible to the change of hours. So as the days start getting shorter after the equinox of summer, this is going to be the signal for them to start cycling. Then they're going to go through the breeding season during late fall, winter, get pregnant, and eventually lamb during the spring and summer, so that these animals are going to have availability of food. So the way that this is presented, and some of my students have seen some of these slides already, is basically through the stimulus in the retinal photoreceptors. So the presence of hours of light is going to stimulate and signal neurologically to the suprachiasmatic nucleus. From here, this information is going to go to the superior cervical ganglion, which is going to produce less or more neuroepinephrine, which is going to go to the pineal gland. The response of the pineal gland to long hours during the day is low production of melatonin. When we have low production of melatonin in the case of the small ruminants, this is going to reflect in a decrease of the production of reproductive hormones. Because of this, these animals are not going to be cycling. As the days start getting shorter, the effect is going to be completely the opposite. The signaling is going to be the same, but this is going to go to the superior cervical ganglion, which is going to increase the production of norepinephrine. It's going to increase the quantity of melatonin that is released by the pineal gland. And this increase in melatonin ultimately is going to increase the release of GnRH in the hypothalamus. And therefore, we are going to have increase of luteinizing hormone and follicle stimulating hormone that are going to be related and basically stimulate the ovary to start cycling. So this is how this happens in the female. So in the female, we are going to have this signaling that is going to create these animals to cycle. But also in the male, we know that there is effect of the season. So we know that The changes in the hours of day and the hours of light has an effect in the scrotal circumference of males. It has an effect in the concentration of testosterone, and it has an effect in the seminal parameters, both volume and quantity and quality. And in some species, for example, if we talk about deer, we will see that this is associated with testosterone, and this testosterone is when these animals are going to grow these big antlers. So this is associated with the breeding season. So based on this information, we can conclude that sheep are seasonal breeders, short day breeders. So this is the time where they are breeding mostly. Having said that, and having made clear that this is influenced by the photoperiod, it's important to realize that that photoperiod is also influenced by other factors. So the location where these animals are is going to have an influence on how deep that anestrus and that breeding season is. So besides the fact that north and south of the equator, you're going to have opposite seasons, the farther that these animals are from the equator line, the deeper the seasons are and the deeper that anestrus is. Fun fact, for example, in Colombia, that is in the equator line, sheep doesn't have seasons. They cycle year round. And that gives you an idea of the influence of those hours of light. Another thing that is important is to understand that different breeds have different genetic susceptibility to these hours of light. So here we have the example of two different breeds. So for example, the Dorset Horn breed starts cycling in July and ends cycling in February. And other breeds like the Blackface, one starts cycling until October and stops cycling in January. So different breeds, different genetics are going to respond different to the effects of photoperiod. So nutrition status is also something that is important, and the production system as well. So based on this information, the research objective is to optimize the breeding season. So the longer this breeding season is, the longer we have the possibility of getting these animals pregnant, the better are going to be the results for two purposes. For once, we are going to be able to improve production because we're going to be able to have products available for a longer period of time, whether this is milk or meat. We are going to have products available year-round. We are going to be able to breed the animals more often. Therefore, we're going to increase the number of lambs per lifetime of ewe. And eventually, this is going to reflect in economical benefits for the producer and for the local economy, in particular for the case of Cyprus. And also, it's important for conservation. So there are breeds and species that are in need for conservation. Reasons why are genetic diversity or just sustainable population of these species. And so these two are going to be reasons why we need to optimize the breeding season of some sheep. Now, a prerequisite to be able to do this is to characterize the reproductive physiology of some of breeds that we are not familiar with. So here I'm going to tell you about two different examples. So let me introduce you to the Florida native sheep. So this is a breed that is considered currently endangered because it has not been paying attention for several years. Nevertheless, it has parasite resistance and is very adapted to the hot and humid climate of Florida. And therefore now there is interest on trying to work with this breed, improve them genetically, and kind of like preserve them as well for the genetic diversity. So the objectives of this research were to characterize some of the unknown reproductive physiological factors of this breed because we were not familiar on when they reach puberty and what was the length of their seasonality. So we came up with a study design where we follow adult ewes and ewe lambs to see when they reach puberty and to find out when they started cycling and when they finished cycling. The way that we did this, we took blood samples weekly for a whole year so that we were able to evaluate the progesterone levels as a way to determine that these animals had ovulated. And then in males, we also follow a group of lamb rams and a group of adult rams to see what were the effects of the seasonality in these males and to see when they reach puberty. We measured testosterone, scrotal circumference, and seminal parameters. What we were able to see in the case of the ewe lambs is that they had been born in the spring of this year. And so here you can see how old they were at the different times. And we can see here as a percentage of how many of them were cycling, that about starting 6 months of age, some of them started cycling, but only until they were all about 9 months in October of that year, they had all started cycling. Starting at the end of November, early December, some of them stopped cycling, which this tells you is that basically you had at this point only 1 month to get these lambs pregnant during that first breeding season, which is not very convenient. The conclusion was that they reach in average puberty at about 8 months. And again, they have a very short breeding period that first year. We continue follow them for the following year. And what you can see here is this is their first breeding season, starting in October, ending in December. But then by the second year, when they are yearlings, they actually started cycling a little bit early, and they match the adult ewes starting cycling in July. This is a different way to present the data. So here you can see how the days start getting shorter. This is the first breeding season, so it started a little bit late because they were too young and it was very short. And then the following year, as soon as the days started getting shorter, we had more than 50% of the animals cycling, indicating that they had reached the breeding season. In the case of the adult ewes, same thing, we were able to determine that starting in July, so very soon after the summer equinox, as soon as the days started getting shorter, they all started cycling, and about early June, 50% of them had cycled, which is what is determined officially like the beginning of the breeding season. So in this case, this animal cycle from July all the way to December, allowing you basically for about 5 to 6 months of a breeding season, which is kind of like what we are looking for, an extended breeding season. In the case of the lambs, we saw that they progressively increased their scrotal circumference, their average testosterone, and so we were able to determine that they reach puberty at about 7 months of age, and that they all passed their breeding soundness evaluation about that time. In the case of the adult males, we were able to see also effects of the seasonality. So we saw that there was an increase on the scrotal circumference, which ultimately is a reference to determine that these animals are preparing themselves for the breeding season. And so although they all the year round had good quality of semen, there was an indication that these animals produce higher volume of sperm and semen when they were closer to their breeding season. So in conclusion for this study, we were able to determine that this breed is indeed a seasonal breed, contrary to what some producers were saying that said that they did not have a cyclicity, that they cycled year round. We determined what was the time where they reached puberty, both for male and female. Some limitations to the study because we use only one herd, and these animals are closely related, and because we did this study only in 1 year, there might be factors that are associated just to that herd and to that specific conditions of that year. What that means is that a lot of this information is crudely applied to this population. Future studies, nevertheless, for the breed, now that there is a characterization of the reproductive characteristics, is that there is going to be possibilities for starting to optimize that breeding season, whether this is the use of hormones or the RAM effect, some things that I will go over in a little bit. So the second research project that I want to show you is with target use. This is a composite breed created in the United States. This study took place in California. We knew when these animals had their breeding season, so what we wanted to do at this point was to improve the period where they were cycling. So we wanted to accelerate the beginning of that breeding season, and we wanted to compare a couple of different methods. So some of the methods that are available for the control of the seasonality is the manipulation with light, so having these animals in a barn where we control the hours of light and the hours of darkness will be a way to do it, basically mimicking the seasons. Another possibility is using the implant, because as we saw, the melatonin is related with the decrease of the daylight and therefore the increase of the reproductive hormones. So if we use melatonin implants, we are mimicking that decrease in the hours of light. And I'm going to go a little bit over the RAM effect and the use of the progesterone priming. There are also two methods that have been used in the past for controlling the seasonality. So in the case of the RAM effect, it is known that the presence of the RAM to a group of females has an effect due to the pheromones of the male, that the LH is going to increase both in the male and the female. As the consequence of that increase in the luteinizing hormone, both male and female are going to start producing more testosterone and more estradiol. Specifically for the case of the female, these increases in LH are going to contribute and accelerate the season, the beginning of the season. And so this is a method that is commonly used by producers. And then the last method that I wanted to talk about is progesterone priming. So basically, the use of progesterone, and actually more than the use of progesterone, the removal of the progesterone removes the negative feedback of the progesterone and facilitates that other hormones increase. And so these devices that you see here are just different commercial devices. These are placed in the vagina of the ewe. They are left for somewhere between 5 and 12 days. And at removal, the negative feedback is going to be removed and these animals are going to start cycling. So we wanted to compare these two last methods, the RAM effect and the use of progesterone. So we used 280 females. We divided them in 6 groups, basically different study groups that combine the different treatments or had just one of them. And then we had a control group. At the end of the experimental period, we introduced the males. Sorry, let me just clarify that these males that we used for the RAM effect were vasectomized rams. So they were there present to produce the RAM effect stimulus, but they were not able to breed them. So at the end of the study period, we put fertile rams with the females and we followed them until lambing so that we were able to see which animals were pregnant earlier and therefore lambed earlier. And what we were able to see is basically here, each of these colors is one of the groups, and each of these decreases basically is equivalent to a lambing. So in other words, these first groups were the ones that started lambing earlier and finished lambing before these ones that took a little bit longer to start lambing and lamb over a shorter period of time. So when we evaluated this statistically, what we found is that the 4 groups that received the progesterone implant were actually reaching that breeding season faster than compared the control and the ones that had only the presence of the RAM. So the conclusion for this study is that in this particular population of animals during this period of time, the seeder was more efficient at accelerating the onset of the cyclicity. We didn't find an additive effect. Therefore, using the RAM and the progesterone did not have positive effect. Just the presence of the progesterone itself had the effect on the acceleration of the breeding season. Limitations, same as before, it was applied for that population only, but it allowed for future studies where we will be able to look at economic feasibility and see if the use of this is economically suitable for the producers. So just as a highlight of the implications and applications of this research, basically we are able to see that there is a relevance in identifying the physiological characteristics of the reproduction of the different breeds, that there is an important in understanding how we can manage product traits so that we can improve the production of these, and that this is going to be relevant not only for production but for conservation as well. And it provides the foundation for future research. That it is for these species that we advance some of the information and the knowledge, or for future studies that take into consideration other breeds or species. So future directions and things that I would be interested on working here in the future will be optimizing the breeding season for production purposes in some of the local breeds. I mean, there are several of these treatments that I mentioned that are already done and used here, but it will be interesting to see how we can optimize this so that we can have a strategic lambing and an increased period of production. Another thing that will be interesting to do will be to try to contribute to the preservation of the genetic diversity with local breeds that have improved genetically during the time here, and potentially other species. And eventually kind of like do a synergetic approach to both conservation and production. So with that, I will just conclude that the production of sustainable products in the sheep industry is very important. We know that in Cyprus it's very relevant as a local income, and it's also important because of a source of protein for the population. And so there is a lot of space for future research that could have an impact into this sustainable production. So that was really fast. I would like to thank you for being here today, and I will be happy to take any questions if you have.